This is Dr. Ahmad, and today we're going to start the new chapter in the urinary system. This is going to be the physiology segment from here on now. We're going to have series of lectures on it, and in this lecture today, we're going to have an overview of the nephrons function, these segments, the different segments that we're going to discuss, the blood supply and the control. Control here means the neuronal control and the endocrine control. From here, we're going to look at the physiology. In the previous lectures, you have seen the anatomy and the histology, but over here, we're going to start looking at the urinary system as a whole from the physiological point of view so you can understand what is the basic functions of it. So there are four primary functions of the renal system. So the very first one is the filtration. So plasma is filtered out from the blood and into the glomerular capsule, and this is the most important function of the kidneys. That's what they're made. The second one is the reabsorption. So materials like solutes, water, ions. So whatever is going to go out, we're going to have to reabsorb it back. The body has to reabsorb it back because not everything is a waste. So whatever the filtrate that goes out in there, something has to taken back for the bodily functions. Then there is a secretion. So not everything is filtered out. Sometime you need even more waste products that needed to be placed into the filtrate. So more metabolic waste products are then placed into the renal tubules, and even more waste products needed to be expelled. And the final function, or the final primary renal function, is the excretion. So the final excreta that includes metabolites, toxins, whatever it is in the body that needed to be expelled, is going to be placed into the urine and excreted from the renal tubules into the collecting ducts, and finally it is drained out into the bladder and out of the body. So before we go into further discussion or the overview of the renal functions, we're going to have to discuss this major concept. It's called osmolality. So what is osmolality? So it is a measure of the number of dissolved particles or solute in the fluid. So that's the basic or the broad definition. So essentially, from the renal point of view, it reflects the concentration of substances like sodium, potassium, chloride, glucose, urea, whatever it is, whatever is placed into that urine. That's what the osmolality is referring to. So from clinically speaking, it is used to evaluate the balance between the water and the dissolved particles in the blood and in the urine. So whatever the difference of there, you have to create the balance of it. That's what the major function of the kidneys. So I mean, whatever the four primary functions that we discussed in the previous slide, the bottom line is you have to create that balance so the body can function optimally. So where does all this primary function happens in the kidney? So the primary function of the kidneys happen in different portions, and depending on all these different portions, they come together into one functional unit, which is called nephron. And there is millions of nephrons in the kidneys. Looking at the gross structure of the kidney, there is an outer cortex, and then there's a medulla. The medulla is divided into outer medulla and the inner medulla, and there's two different type of nephrons present depending on the location. They are cortical nephrons, which is, of course, the name suggests, in the cortex, and finally there is juxtamedullary nephrons, which is just at the border for the outer medulla, inner medulla, and cortex. So those are important as well. So then there's the final portions where all these primary functions can happen in the nephron segment. So each and every segment has its own set of functions, which we are going to discuss into the further details. But in this lecture, we're just going to look at the overview of what these functions are. So over here, this picture is basically the anatomic component of the kidneys. This is the osmotic component or the physiological component of the kidneys. So we have discussed that time and again into the renal anatomy in the previous slides. We have also discussed that in the renal histology point of view as well. You see the renal artery going into interlobar artery, arching up, making an arcuate artery, interlobular artery, then making the afferent efferent arterioles and different capillary beds, glomerulus, etc. So over here you see cortex. This is the medulla part. There's an outer medulla, inner medulla. Then the kidneys are covered by the renal capsules. All these medulla, they have an apex. This is the pyramidal shape. The base of the pyramid is up against the cortex. This is the apex is renal papilla right here. Renal column is basically the divisions between them. We have the minor calyx. Two or three different papillae make minor calyx. Two or three different minor calyx make a major calyx, and they make the renal pelvis right there. And finally, it gets into the ureters right there. But looking at from the osmotic components. So osmolality, as we have discussed that in the previous slides, the cortex has the lowest osmolality, while the inner medulla has the highest osmolality, and they can vary between 300 to 1200 milliosmoles. So lowest, as we discussed the anatomical portion, cortex, outer medulla, inner medulla. Of course, we have discussed that. We have seen that in the previous slides. The lowest of the osmolality is 300 milliosmoles, and that is in the cortex. The highest osmolality is 1200 milliosmoles, and that is in the inner medulla. And in normal condition, this actually determines the concentration of the urine. So, under special circumstances, the osmolality can drop below 300 milliosmoles, but that is under the special circumstances. 300 milliosmoles is the minimum below. So let's look at the nephron segments now. So there's two different type of neurons. Of course, we have discussed that: cortical nephrons, juxtamedullary nephrons. So one major difference is the loop of Henle. So in the cortical nephron, it mostly the renal tubule, when it makes that loop of Henle, it mostly stays in the cortex part. However, it can also traverse down a little bit into the outer medulla, while in the juxtamedullary nephrons, the loop of Henle, it traverses deep into the inner medulla. So depth of medulla is important in determining the osmotic gradient. Juxtamedullary nephrons can reach in the higher osmolality portions in the medulla, and these nephrons allow us to better concentrate the urine, which is important. So now let's look at the nephron segment. We have discussed that before, but here let's look at from the physiological point of view. It's just an overview. So we have a Bowman's capsule or glomerular capsule. So major process, the filtration occurs here. The small amount of plasma is filtered out, which travel to the proximal convoluted tubule. The next one is the proximal convoluted tubule. So the major reabsorption happens here, and it has a brush border or the microvilli or the invaginations into its walls, which increase the surface area, which in turns will have a major reabsorption that happens here. So it is majorly present in the cortex, and it has a low osmolality of 300 milliosmoles. So basically you can say it is isoosmotic. Then we have the proximal straight tubules. They are also heavily involved in the reabsorption. However, the structure has a lesser invagination than the proximal convoluted tubules. The next portion of the nephrons is the thin descending limb. It varies in terms of length depending if they belong to the cortical nephrons or the juxtamedullary nephrons. So the primary purpose is to allow water to move out. Then we have the hairpin turn, after which is also called as the loop of Henle. And then the loop of Henle changes into the thin ascending limb. Thin descending going down and thin ascending going up. Between that, there is a loop. So thin descending and ascending limb along with the loop of Henle are basically called thin because they are structurally thin. They're involved in the medulla's involvement in the osmolality to do most of the work. So the osmolality over here is 1200 milliosmoles. It started to increase from 300 going down to 1200, and when it goes back up through the thin ascending limb, it started to decrease again. Then we have a thick ascending limb. So it has a function of transport material, and it requires energy to transport from the apical membranes to the blood. Finally, we have a distal convoluted tubule. This distal convoluted tubule is located a bit higher, and it is also mostly present in the cortex. So the osmolality here is again 300 milliosmoles. You start with 300 from the glomerular capsules to the proximal convoluted tubule, go down to 1200 into the medulla, go back up to 300 again. Or sorry, I mean from the number point of view, go down to 300, but from the gradient point from 3 to 12 and 12 to 300 again. So the location of distal convoluted tubule has to do something with the maintaining the osmolality as well. The final nephron segment is the collecting duct. So it is the area of high osmolality because it starts from top and goes down deeper into the medulla and out into the papillae, minor calyx, and again. So the osmolality increases from the cortex into the medulla. So collecting ducts is present, of course, in high osmolality, and by utilizing this inherent osmolality, so if there is a channel available, it can allow the water to be transported across. So its major function is to transport water, and usually occurs with the presence of certain hormones. So now we have just looked at the tubular functions of the nephrons. Now we're going to look at the blood supply. We have, depending on the location, of the nephrons, there will be capillary beds, and they will perform the functions of the reabsorptions. So up until now, we have discussed in the histology and anatomy how it works. So we have the afferent arterioles. The blood comes from interlobar artery, arcuate artery, interlobular artery, which divide further into the small arteries, which goes into the nephrons. So the first part is the afferent arterioles, which changes into this capillary tuft, which is called glomerular capillaries, where filtration happens. Then we have the efferent arterioles. So after that tuft changes, it goes out into the efferent arterioles. So from here, there could be two different capillary bed depending on the location of the nephrons. We have already discussed that in histology. Let's look at that again. So the first capillary bed is the peritubular capillaries. So this is, again, you look at the kidney and we're looking at the cortex. So this is the renal again. So this is the afferent arteriole going into making the glomerulus, and going out is the efferent arteriole. So these yellow convoluted structure going up and down, that is the renal tubules. You see the blood vessels, it's changing. This is the peritubular capillaries. So this is present in the cortical nephrons and covers the major structures there. So that include the proximal convoluted tubule, proximal straight tubule, the distal convoluted tubules. All of these structures are present in the cortex, and these capillaries are present for the structures in the low osmolality areas. Now there's another capillary bed that we have to look at, if depending on the depending on the nephron location, and this capillary bed is present to reabsorb the materials from the medullary nephrons, and it is vasa recta. So as the renal tubules here are plunging deep into the inner medulla. So over here you see two different nephrons. So you see this is the cortical nephron. You see the loop of Henle, it's traversing a little bit into the outer medulla. But if you look at the juxtamedullary nephron, you see the loop of Henle is going deep into the inner medulla. So this capillary bed is called the vasa recta, and they have a unique feature to deal with the high osmolality situations. So what are the blood supply? So how the various segment of the kidneys and blood flow is controlled here. So the first component that actually control is the neural component. So there's two. So so the two components again is neural and endocrine components. That's how the blood supply is controlled. The very first one is the neural component. So the neural component for the control is from the sympathetic nervous system innervation area. So how does that affect the blood supply? So it controls the blood supply or blood flow through controlling the afferent and efferent arterioles. It controls the blood flow through the glomerular capillaries, and it can control either one, means either afferent or efferent or both of them. And they will also affect the glomerular filtration rate, which is also an important topic. We're going to discuss that in the later slides. So sympathetic nervous system is going to signal the small cells in the juxtaglomerular cells, which release renin, and renin is going to activate the renin angiotensin aldosterone system, which then controls the sodium level that will be within the urine. So now let's look at the endocrine component. So there's three different hormones or endocrine components of the renal blood supply system. The first one is renin. Of course, we have discussed that from the previous slide. Renin produced from the juxtaglomerular cell. If you want to know what the juxtaglomerular cells are, please review the histology slides. It has been discussed in details over there. So produced from the juxtaglomerular cells for the blood pressure control, of course, through the renin, angiotensin, and aldosterone system. The second one is the release of the erythropoietin. So it is produced from the cortex and outer medulla and signals the bone marrow to produce more red blood cells. So this is one of the component. Then there is another one, it's called the calcitriol. It is produced from the calcidiol, which signals the increase of the absorption of the calcium ions from the intestines. It also increases the tubular reabsorption of calcium, and it also releases the calcium from the bone, and this hormonal regulation then work for turning the form of vitamin D into its most active form. So it has its control into the vitamin D production or activation. So as I discussed before, the basic bottom line is to maintain the bodily functions, and maintaining the bodily function is called homeostasis. So with all these function overview that we have seen, what it actually accomplishes. So the very first one is the blood pressure control. We have already seen that in sympathetic nervous system. The sympathetic nervous system is going to innervate that afferent and efferent arterioles. And of course, if you remember from the histology, the afferent arterioles, they have this smooth muscle cell, so it can decrease or increase the diameter. And those smooth muscle cells are also called as the juxtaglomerular cells, which will also signal to release the renin. The second one is the electrolyte and osmolality control. So it is going to control the blood volume. Then we have the water volume control. We have the glucose metabolism, which is important here, especially in, you're going to learn that in the biochemistry slides later, gluconeogenesis. Then we have the oxygen carrying capacity. You can ask, how does that work? So it is going to indirectly affect that by stimulating the red blood cell production through erythropoietin. Then we have other electrolytes control and clearance, metabolites, etc. For example, if a toxin or a drug metabolite is filtered out and urinated, so that's how it clears it, and that's how it filters it out. And the last one is the acid base balance. So sodium bicarbonate production and hydrogen ion balances, it's actually controlled via kidney. So we're going to stop here. This is the overview of the physiology. There's going to be many more slides from the physiology of the urinary system. So stay tuned and I'll see you soon. Thank you so much for your support.